It's my great pleasure to introduce Yuhan from, I guess he's working at the startup Neptune Bio now, but he got his PhD in Rahul Satij's lab at New York Genome Center. And he's done some awesome work across multimodal learning and scaling single cell sequencing based analyses. And he's done a lot, one of the main developers for Seurat, which I guess many of us have used. So yeah, I'm really excited to hear your talk today.
Okay, cool. Right. Thank you for your introduction, and thank you for your invitation to be here. And like Salih introduced, I just finished my PhD training in Rahul Satij's lab at NYU and New York Genome Center this summer. And also I'm the lead developer of Seurat and latest 2 versions. Today I'm going to talk about our latest work, Seurat v5, about the cross-modality mapping and some large-scale clustering of the single-cell data. But before that, right now I'm a co-founder and equity holder and employee at the Neptune Bio. But just before we talk about the software Seurat, let's just look at these 3 paintings briefly, right, to see how the focus of artists have evolved across ages. Right, you can see in the early 16th centuries, right, the artists from classicism, they emphasized on the perspective drawing and the human anatomical accuracy. You can see their goal is to get a faithful representations of the human appearance. And then in the 19th century, the Impressionism took the center stage, right? So this movement shifted the focus towards capturing the fleeting moments of light and the color. And then in the early 20th century, the Cubism emerged with this fragmented abstract forms, just gave a new trend that was a multi-view representation. So no longer satisfied with a single viewpoint, the artists there attempt to picture the subject from multiple perspectives simultaneously. And they believe that this way they can, capture the inherent characteristics of the subject more effectively. But why I talk this? How is this related to cell? Do we go to a wrong seminar? The thing is that, those milestones in those paintings kind of mirrored very well of how we understand the cell, right? While Hooke first created the microscope, we began to study the morphology of the cells. And then because of the RNA probe and the fluorescent antibodies, and we can study the genes and the proteins in the cells. And right now we have those fancy multimodal technologies. We can simultaneously study the multiple aspects of the cell, right? We can capture more comprehensive understanding of the cellular process. It seems to be a coincidence, but actually I think it's more likely that, the technology advancement always, reshape our understanding of the subject that we study. Okay, let's go to the topic today, right? The single-cell genomic sequence. As we all know, right, the single-cell RNA-seq technology, for example, like DropSeq, right, allow us to massively parallel profile gene expression of thousands of cells. Then we are going to get a matrix like that. Unlike the conventional methods like PCR, Western blot, or even the FACS plot, it just takes minutes to interpret the data. But you can see to interpret this type of matrix, it's not a trivial. We need some specific algorithm to process this type of data. That's why, this thing is kind of motivated Rahul at that time as a postdoc in Broad. He created the first version of Seurat. As we all know, right, scientists are not satisfied with current technology. We're always seeing those new fancy technology there to give us better understanding of the cells. And then, also motivate us to create a new different version of Seurat, also have a new statistical models to help people, interpret those data better, learn more about the biology. Today, I will mainly I will also introduce this human cell atlas. And we are pretty familiar with that. And right now, not just this, also some other different consortium like the CZI and HubMap. We have a lot of atlas right there. And they have the good annotations, not just those big organism level. On different tissues, we also have those high quality annotations there. And then the thing is that because of those resources, it also motivated computational biologists to come up with this single cell reference mapping algorithm, very similar to the genome reference mapping, right? So we have a good annotated stuff. Let's find a way you effectively transfer those built information into the new query cell. And in our Seurat v4, we also have our own single cell reference mapping. I also like to build a web app called Asmus there. So right now we have 12 reference for different tissues. And you can see there are over 10,000 data sets, over 400 million cells mapped to that app. It's good, but the problem is that all this just for the RNA-seq, right? If you have your own ATAC-seq or histone modification at a single cell level, you cannot use this reference at all. That means we have to build a new series of those references again and again when we have a new modality. It's just literally impossible. And then you can see in Seurat v3, right? Team Stewart from our lab, he figured out a way to map the single cell ATAC-seq onto the single cell RNA-seq. Basically, that just take the gene body sum of the ATAC signal as a proxy for the transcription output, like the gene activity score. Then they are in the same space, then they can put them together. For example, here is the single cell data from the Morse cortex. You can see generally the TAC cell and the RNA cell integrated well. The cells from the same cell type, they are kind of in the same place. But this type of a transformation requires a strong assumption that may not hold true all the time, particularly on the transitioning or those developmental systems. And then we know, right, we have those single cell multiomic technologies allow us to measure multiple modalities simultaneously at the single cell level. Those kind of the measurements we get, it's just like the biological translator. When we know one modality, we can just check the cells. We know what the other modality looks like. And this provides our opportunities to say, okay, if we can use something to connect those unpaired modalities. So here we propose our methods called bridge integration that leverage this multiomic data set as a molecular bridge to connect those two single modality data sets. The good thing is that, for us, we don't need to worry about, if there's a path, can go through all the modalities and only cross 7 bridges once, that bothered Euler 100 years ago. We only care about, 2 modalities and the 1 bridge in the middle. And let me first quickly introduce the dictionary learning. That's a key concept we're going to use today. The key idea is that, if we can reconstruct the object, right, based on a weighted sum of the little elements there. For example, we take a noise image, then we can just get the little patches of the image. Then our goal is to use those little patches to reconstruct this noise image. Right? If we can do that, then you can see the dimension of the image is reduced from millions of pixels to those hundreds of patches. Then during this reconstruction step, we can easily denoise the image and also compress the image. So this technology is commonly used in the image analysis, but not really in the genomics. So you can see the key ideas, the most important thing is that represent the data set as a combination of individual elements. And those elements come from this image itself. It also comes from a collection of other high quality images. But how the dictionary learning can solve our question? Our question is that how we can map query to the reference when they measure different things, right? For example, here, our lab built a good single cell bone marrow reference. And you can see particularly for those HSC, those developmental trajectory, it's nice. We put a lot of efforts there. And there we have a single cell ATAC-seq from Greenleaf's lab. And we want to map them to this reference. But we cannot do that because those single cell ATAC-seq measure peaks, the reference is on genes. Now, how can we solve this? So that's where the dictionary learning comes in. So our intuition is that, okay, because we know we have those multiome cells, if we can use those multiome cells in the middle as a bridge to connect these 2 modalities. So you can see the thing we're going to do is that we will treat those multiome cells just as those dictionary elements. For example, we know, right, the multiome cells, they measure the gene expression, and we can use those multiome cells to reconstruct the gene expression profile of the single cell data. Because they measure, both of them measure the genes. And we can also use multiome cells to reconstruct the single cell ATAC-seq, because both of them measure the peaks. Right now, the trick is that you can see that both modalities are reconstructed by the same set of cells. Right now, those 2 things, those 2 types of cells, in the same space, in this multiome cell space. And then we can, transfer labels or do those trajectory analysis jointly. Right? And just let me go through a little in the detail of how do we do that. So here, this X is just the RNA gene expression matrix. Each row is a cell, column is the genes. And here is a multiome matrix, right? So goal is that we want to find a matrix D. This is unknown. We can see, how can we do this row combination of the MX? You can see the row here is the multiome cells. How do we do the row combination there? We can reconstruct this matrix. Then our goal is to learn a matrix D is a dictionary representation of those single cell data. You can see the dimension there. Each row is a single cell RNA-seq cell, and column is a multiomic cell. And we can do this for both RNA-seq and the tag-seq. Then we can have those two representations. They are in the same space. But you may also notice that these two matrices, they have a problem. I mean, one, we have thousands of multiomic cells. These two matrices are not compact. They are very huge and dense. It's not good we calculate and or store this type of matrix at all. Then we think, okay, how can we decrease the dimension here? Our first idea is that, okay, if we can aggregate this matrix just based on their annotations, right? Maybe just based on if they are all the T cells, we can aggregate a little bit. Or if we can do this based on their clustering results. But then we realize, oh, if we do that, then all this bridge integration will highly dependent on how do we annotate those bridge data sets. Then we think, okay, if we can have a more continuous way to smooth this matrix or decrease the dimension. Then one idea came to us is that, okay, maybe this Laplacian eigendecomposition of a graph that can be used here. Because the good thing for those Laplacian eigendecomposition is that it's a low dimensional representation that can maximally keep the neighbor structure of the graph. Then, because those neighbors in the graph, that is the basic unions. How do we define the cellular clusters? Then we think, okay, maybe we can try that. So we do the Laplacian eigendecomposition of the bridge of the SN graph of those multiomic data. Then we can have this low dimensional representation, this UL. Then we project this both the dx and dy into that space. And then when we do that, you can see the dimension of those cells. They are shifted from the thousands of multiomic cells into those 50 Laplacian space. Then right now they are in the low dimensional space, in the shared of the low dimensional space. Then we can transfer labels and jointly analyze this. Also do the batch correction.
I have a quick question.
Yeah.
So you construct this graph on the multiome cells, which you're using to do the dimension introduction.
Yeah.
How do you weight how each of the
modalities? Yes, that's a good question. Yes, thank you. Yeah, and actually that is our main focus in our Seurat V4 paper. It's mainly we work on we have a method called weighted near neighbor graph. That is mainly a clustering algorithm and try to integrate. That is mainly for the single cell multimodal data. We try to find a way to effectively integrate those two modalities to get one representation. So the key idea is that basically we calculate for each specific cell, we calculate the modality weight for those RNA and the tag. Then we say, okay, which modality have more information there? Then we have a new distance metric, and we construct a single representation for both modalities. And then we can have SN graph there, and basically this wide from that.
I have a quick question again about the dimensionality reduction. Can you explain some intuition for why the Laplacian eigenvectors are better than, for example, principal components?
Yes. It's a good question, right? So as we know, right, the principle, the PCA, is mainly to explain the variance of the matrix. But like I mentioned, the key stuff we try to do is that we try to group the cells together. Then we think, okay, how can we find low dimensional representation to represent those neighbor structures, to represent those similarities things? Then we realize, okay, this one is better because we don't care much about the variance in that, in those cell similarity graph. Yeah. So here we have an assumption actually is that a bridge cells and their neighbors contribute similarly in this dictionary representation. Yeah. Okay. Yeah, when we put them in the same space, right, you can see we can transfer all the labels from the RNA-seq to this tag-seq. You can see all the efforts we did to refine annotation in the RNA-seq can be used to annotate our new additional query data. You can see then when we have this bridge, it just open up a new area. Then we can map all those different type of tag-seq using this RNA reference. And here I want to highlight one result is here. It's just one thing called ASDC. It's a very small populations. And this population was discovered, Rahul contributed to discover this population when he was postdoc here. This is a rare type of dendritic cells. It has two markers, cyclic 6 and the AXL. It was first discovered by the single-cell RNA-seq. Right now, because it's bridge integration, we identify those cells from the single-cell ATAC-seq. Right, so you can see in this, there is just a unique enhancer that only enriched in those ASDC cells from the ATAC-seq profile. The thing is, because it's very rare, without this, supervised way, this mapping, there's no way we can identify those populations from the single-cell ATAC-seq alone. And beyond this, the cell type transferring, one of the cells they are in the same space.
For the previous one, would you require that the multiome also capture the extremely rare groups?
Yes. Actually, in the multiome, there are just a very few ASDC cells. We find, when this population have strong signal, even though you have very few of them, that's still enough. Because the multiome, they have the RNA-seq profile, so they have the signal there.
Yeah. Can you just go back to the graph Laplacian? Could you just explain what the G matrix is? I just didn't quite understand how the experimental data goes into what the data are.
Sorry, what do you mean G matrix?
G, capital G.
Oh, yeah. So this capital G, that is shared nearest neighbor graph of the multiome data. Like I mentioned, our weighted nearest neighbor methods, we can create a multimodal graph that represents both modalities. So, and also this graph, we only need to calculate once. Because when we have new query data, we only need to calculate this LY first. When we build the bridge, when we connect the RNA with the bridge there, all this calculation just need to perform the once.
So what is N multiomic? This is the number of
multiome cells. Yes. Yeah. Thank you. Yeah. And you can see right now the ATAC-seq and RNA cells, they are in the same space. And then we can perform some, for example, here, this is a myeloid differentiation trajectory. We build using both RNA-seq and ATAC-seq cells. So here we're not smoothing them. You can see those individual cells. Here are the RNA cells, here are the ATAC cells. They have those different gene and ATAC-seq markers. While right now they're in the same space, we can just we can do this pseudotime analysis for both RNA and ATAC. The thing is, right now they are in the same pseudotime scale. So for example, here we identify some genes whose expression lags behind the chromatin state. That means, those genes, their chromatin states, they open first, and then we see the increase of those genes. And you can see that is example, then if we directly convert the ATAC peaks to the gene activity score, we will totally missing all those signals. And interestingly, you can see all those genes actually are cell cycle related genes. Yeah, so the example I showed you is just that single cell and ATAC integration. But this bridge integration is a general strategy. It's just when you have the different bridge in the middle, there can be this single RNA reference can be used for all those different type of things, like the histone modification or DNA methylation or even the cytoph data. So here I just want to show you one more example that's about the single cell DNA methylation. For example, the left is human brain data from Acker's lab. They sequence in a single cell DNA methylation data. It's good we have all those different clusters, but the DNA methylation data is a little bit harder to annotate those clusters. So they just have, you can see a lot of things, they just -1, -2. And on the other side, our lab built collaborated with the Allen Brain Institute. We kind of refine those annotations of the single cell RNA-seq data. So we believe that those high quality annotations should be helpful to annotate the single cell methylation. And here we use SNC2T-seq. That is the multimodal data can measure cDNA methylation and the single cell and the RNA expression simultaneously. And then this is the mapped result. You can see here we are not finding any new clusters. But the key thing is that here we label those DNA methylation cells with a defined ontology. Instead of call those cluster 1, cluster 2, we use those RNA-seq can help us, to give them a name that can be used consistently across different modalities and across different samples. It will be much easier for us, to do the comparison analysis. And so this is just a summary about the bridge integration. So you can see here we can use the multimodal data as a molecular bridge. And this is a general framework. When you have a bridge to connect a new modality, then this bridge integration methods can work there.
Yeah, just about the Seurat. So is the is this bridge integration and all the reference dredges, is that built into the package? So
yes.
I just run with.
Yes, exactly. You can find all the variants in our website. Yeah, I'll show you where you can find them. Yeah.
Yuhan,
yeah.
I missed this, but can you just re-explain how you learned the dictionary representation?
Oh, sure. Yeah.
I just think I didn't quite.
Basically, that is just the reconstruction of the expression, right? You can see this is the RNA-seq gene expression profile. This is a multiomic gene expression profile. We try to learn a matrix DX here to reconstruct this. Yeah. And beyond this, except for this cross-modality integration, the other one direction that people put a lot of efforts for the new technology is the throughput of the single-cell RNA-seq. You can see before we can just measure tens, then we get hundreds, thousands. Right now, those SCAR-RNA-seq and Cypher-RNA-seq, you can easily get millions of level of cells. Then because of those the expansion of those data, the most computational methods become inefficient, either requiring huge memory or takes a very long time to run. That's why then we think, okay, how can we solve this problem? And here I want to use one text data as an example. This is a 1.3 million data set from the mouse embryonic brain. It was produced, 2017, so I have a period of time. Then you can see text show the brute force way to clustering this data. It takes, it took 350 CPU hours and 300 gigabit memory. It's a huge resource. Most labs don't have this resource to analyze this type of data. Then we think, even though, okay, this time we can do this in this way, but right now we are facing the tens of millions of data. Maybe in the future, we are going to handle hundreds of millions of data. And we cannot just buy bigger and bigger computers. Then just we need a fundamental different algorithm to solve this question. Right, so one idea we can use is that, okay, we think, okay, if we can get some representative cells first, we can sketch the matrix. Then we do those computing intensive stuff on those representative cells, those small subsets, and then find an efficient way to propagate the information from the sketch one to the full data set. Right, so in the metric, let me just paraphrase the question in this metric way. So this A is our gene expression matrix. Right? Our goal is to find a sketch matrix S. The S, you can see each row is one hot vector. And the length of the row is equal to the number of cells in this matrix A. And the one indicates the position of what cells we select from that matrix. So our goal is that, okay, how can we build a matrix S that can get those representative cells? Well, the first question is that, okay, what the meaning of the representative? So we are inspired by the work from Brian and Bonnie, and you can see that is there. How do they define this representative? So they have a method called geometric sketching. Their goal is to sample the cells that are evenly across the geometric structure of the transcriptome. And there they can maintain the rare and abundant populations. Also, when you do this, it doesn't require a pre-clustering of the data set. That's important. And also, it doesn't depend on the extensive computational resource. And this also inspired us, but we started from different aspects trying to solve this similar question. Right. The thing we start is that is from the gene covariance matrix. Because the gene covariance matrix, we know they describe the relationship between the gene and the gene. And let me take an example here. So this is a gene covariance matrix from a single cell bone marrow data. Right, so here, each row and the column, they are just the genes. And right now here, I just group the genes based on the markers for different cell types. Right, for example, here, this is the genes for the T cells. They show the similar pattern there. And when I remove the T cells from this data set and calculate this gene covariance again, you can see we don't see a strong patterns for those T cell genes anymore. A good thing for this gene covariance matrix is that no matter how do we sample the cells, then this matrix, the space there is fixed, right? It's just look at the gene, so it's not related on the cells. So that's why, so here, and also it reflects, right, the gene covariance matrix can reflect the cellular structures in this data set. And then, so.
I have a question about that.
Yeah.
So do you think for any cell population, it has a unique kind of block in the gene covariance?
Maybe not a real unique block, but they should have some signal. Because we said they are unique population, that means they have some unique gene program there.
So you would say that, one characterization of a cell population of interest could be
Reflected.
definition.
Yes, some gene space, right? Just like when we have a matrix, we can do the SVD. It's one matrix related to genes and one matrix related to cells. They are not independent.
I guess what I'm asking is, think about defining the population in terms of the mean, or having some unique, but you're saying here, I guess you're not looking at the mean, you're looking at the covariance.
Yeah.
You're saying that actually it should also have a unique covariance.
Yes, because we define the population, we define those cell types among these samples. Then if they are unique, then they will show some variance there, right? Yeah. But if it is a pure one population, then we cannot see this covariance. Yeah. And okay, then this is how do we calculate, assume, right, this matrix is centered and there, with just the easiest way to calculate the gene covariance matrix, right? For example, here, this is a matrix A, the row is a cell, the column is the genes. We can do the SVD there. Then we calculate the gene covariance matrix, just A transpose A. This is by the gene matrix. Then we get that V sigma square and V transpose. And then we can do the same thing for the sketch data. Remember, this S is the sketch matrix, and this SA is the sketch version of that. Then we can calculate the covariance for the sketch cells. So then we can get a result there. A little bit complicated. Then our goal is that we try to minimize the difference between the covariance of the full data and the sketch data. Then we can just plug into the SVD forms to get that result. Then we look at that, okay. Then we notice, right, in this case, only S is at here.
Sorry, what's the motive of developing the new method on top of the GeoSketch?
Oh, thanks, yeah. So the thing we noticed that is that also because the geosketching, they also require the PCA step in the middle. And we noticed that, we will show you later that right now you can see we still need to do the PCA, the SVD. But later we realized that there is a more efficient algorithm. We can calculate this leverage score fast. Yeah, sorry, let's still go back here. Right, you can see in this construction, right, this V and the sigma, they are fixed. Right, the unknown thing is just the S, because the UV sigma is just from the SVD of the matrix A. Then we just put the V sigma from outside in the middle. We have the I subtract U transpose S transpose SU. Then the thing we noticed that, right, based on this Cauchy-Schwarz inequity stuff, we can just separate those V sigma from outside. Then our focus is just the matrix here. But instead of directly minimize the difference there, here we can also minimize the upper bound of the difference. Then the key question is that, okay, how can we find a matrix S that can minimize this error? And the good thing we notice, okay, this is a pretty classical question in this randomized linear algebra. And they know that, okay, when we sample the cells, when we sample the S matrix, based on the row sum of this metric U, based on this probability, then they can get a constant upper bound of this matrix. And also the expectation of this one is equal to 0, and the variance of this loss, if we sample this way, is also show they are the minimal variance. And but the intuition is that, okay, how can we construct this S matrix? If we sample the S matrix like that, this part is similar to this identity matrix. Then if that part is equal to the identity matrix, then you can see this will be the V sigma square V transpose. Then these two parts are approximately equal to each other. But like I mentioned, it seems we need SVD to get this population. But the good thing is that we also find a different paper, the Woodruff and David, they come up with a new method, a more faster way to calculate this leverage score. Here they use the random projection and the random embeddings to increase the speed. Right? Let me just briefly talk about how they implement this algorithm. So here you can see we write as random. This random is not the sketch matrix we talked before. It's not one hot vector. It's just a pure random matrix. You can think it is just from generated from the Gaussian, but we can also find a more discrete way to generate this pure random matrix S. And use this S times in the cell side of the matrix. The goal here is that because they want to learn some gene structure there, then they think, okay, we can, based on the Jonathan Lindstrom lemma, when we can randomize this cell space, those gene relations are still kept there. So then the thing is that first, let's see, when they times this S with A, this matrix becomes small. Then the dimension is S times D. The S is pretty small, they can just put the 5,000, assume this N is 1 million. And instead of do a QR decomposition, it's 1 million times D, we can do a QR decomposition on this time S times D. It's 5,000 times 2,000, assume. And let's assume just D is equal to 2,000, we focus on those variable Gs. Then we can efficiently get this Q sketch and this R sketch. So the goal here actually, this R, let me write a little bit down here. So they try to avoid is that, when we have a matrix A, we can do a QR decomposition, right? This get a Q and get a R. But to this QR decomposition, that is expensive because this matrix is huge. The cool thing is, yes, the Q is orthogonal basis and the R is this upper triangular matrix. Why they want to do this? Because when we have orthogonal basis, we can get the Lagrange score. Because let's see, the definition of the Lagrange score is the row length of the U vector. That is our orthogonal basis for the cell of the original matrix A. So they don't want to do this, but they still want to get this R. Because when we do this A times R inverse, we can get the Q. So if we can approximate this R inverse, then we can easily get a orthogonal space. Then they show this R sketch. It's similar to this R because this S is a random matrix. Then they can use this A times the R inverse sketch that is similar to our orthogonal basis. And this one is also the well condition. That means the maximal and the minimal singular value, they are similar. And still, this N times D space is still big because D, for example, like 2,000, still want to decrease the dimension. Otherwise, the calculation still takes time. Then they put another random matrix, the Jonathan Lindstrom embeddings, to further decrease the dimension. Then they can get this matrix Z is N times N. This N is a million, and for example, just like 200. Then they just calculate the row sum of this matrix. They can get the Lagrange score for each individual cell. Okay, let me show you what does this look like. Then it will become more clear. All right, for example, here I took a PBMC 33,000 data from the TANKS website, and then we calculated the Lagrange score. The cool thing, right, you can notice that the cells from the small population, like HSC or plasmablast, they have the high Lagrange score. And the cells from those abundant population, like naive cell, they have a low Lagrange score. When we calculate the Lagrange score, we know nothing about the clustering or cell type annotation. The only input is the count matrix. And when we can average, each cell type based on their Lagrange score. So here on the plot there, each dot is one cell type. The x-axis show the size of that cell type. On the y-axis show the average Lagrange score there. And here we see an inversely proportional relation between the population size and the Lagrange score. So that means when we sample the cells based on the Lagrange score, we can get a more balanced populations in the sketch data set.
So this data, I guess intuitively is saying something like, how does the Lagrange score of a cell relate to the density
of the cell?
So density
estimation. Yeah. The Lagrange score is originally used for the outlier detection. The thing is that, because those outliers, when we remove the outliers, there's a big change in the gene covariance matrix or the feature covariance matrix. And that's something, that's why they can capture those big change.
Yeah, I guess my question is about one way that you could do the sketching, I guess, is like by doing some density estimate.
Yes.
And then doing downsampling
based on
density.
Yeah, inverse the density.
Yeah, inverse. Yes. Yeah. But I guess here you're using the Lagrange score as something. So do you and you're saying that it's kind of similar, like the very low density cell types.
Yes.
Have the,
yes.
So somehow these seem related. related?
Yes, because you can think that, right, for example, the cells from the naive T cells. When we remove some naive T cells, the gene covariance matrix did not change at all because all the signatures are still there. But for example, like HSC, if we totally remove that, then all the marker genes related to HSC, they will lose the signal in this covariance matrix.
It's not just density, it's density in combination with effect on the
signal. Yes, yes. The unique gene program. Yeah. And right, you can see, but this one is still pretty small. Then we try this leverage score on much bigger data set. We have this here, we have 2 million cells. It's from J. Chandri lab. And it takes around 10 minutes to calculate the leverage score of this 2 million cells. And it will take hours to really perform a PCA on this huge data set. And right, we know, right, when we do the single cell analysis, it's not just about clustering, we also care about those batch correction. And here we collected 1 million long data set from the community and across 11 public projects. And yes, we have so many cool, good, batch correction algorithms. But if we directly use those algorithms on this community level data set, it will become inefficient. Then we say, okay, if we can do a similar thing or to improve the efficiency of this process, then what we are going to do is similar. For each data set, before we do the batch correction, we get a dictionary. We use a leverage score to generate a dictionary for the specific data set. And then we can do this reconstruction, get an index to see, okay, how can we use this dictionary to reconstruct the data set one? And then the dictionary data set, they are pretty small. Then we use the batch correction methods to batch correct those dictionary cells. We get a batch corrected dictionary cells. Then we use the same index we built here to reconstruct the batch corrected gene expression profile for all the cells. So you can see those computational intensive steps we applied on the dictionary, therefore use just an efficient way to propagate back to the full data. And that is our rationale. You can see this is the results we did for those 11 long data set. And it takes around 55 minutes to integrate this 1 million cells. And yeah, I think it's pretty fast. If we use at least Seurat, it will take over 24 hours to integrate this 11 data set. And here also I want to highlight one result, this allocyte. This is a very rare, super rare population. It's around 0.04%, in the lung. And people know that these genes have many expression, many cystic fibrosis related genes. And this population is largely ignored before. But just because but right now in our sketch integration, we can see they have those strong markers, they form our own unique clusters. That means our dictionary learning, we are not going to lose those rare populations. The reason we didn't lose the population is that, based on this 0.04%, if we sample 5,000 cells from there, we can only get 2 cells, right? If we sample them uniformly. But you can see the population there, they have very high leverage score. So when we sample the cells based on leverage score, you can see amount of 5,000, we can get 40 element sites. And then they are easily to form clusters in those dictionary, those representative data. And right, sorry, yeah, like here, still we talk about 1 million. What's the limitation of sketch integration? Then we just try to say, okay, how big data set we can play. And in this year, the J. Chandri lab have a new data set. That's a single cell transcription atlas of the mouse embryonic from the late gastrulation to the birth. Right now, here they have over 11 million cells. And then here we just put the sketch integration, sample 100,000 cells from that, and we learn a PCA space. Then we just project the entire those 11 million cells onto that space. So you can see this is the UMAP visualization of those 11 million cells. And here you can see the annotations are from the original paper. You can see here we keep both the major cell types and also maintain those super rare populations. And the most striking thing is that in here, we can visualize the real-time developmental trajectory because they keep track of those developmental stage of those cells. And we see the gradient, right, on this UMAP here. And also very interesting, we did observe, right, this multiple lineages of those HSC populations. And that just means that, the leverage score sampling, we can maintain the biological signals, not just the big and rare populations, also those, the developmental trajectory stuff. Yeah, then just recently also the CCI organized census data. They try to collect all the available human single cell data. They have 33 million cells. Then we tried our sketch integration again. Yes, it seems we not reach our limits. So this is unsupervised clustering of those 33 million cells. And yeah, so this is just a pure unsupervised clustering. Then we can compare our cluster numbers with their stored cell type information. Generally, right, we get a good agreement there. And right, some low resolutions, for example, here they indicate the neurons. That one corresponds to the multiple clusters. So the point is that you can see the sketch integration methods, they maintain the cellular signal in the original data. And right now, we haven't reached our limit there.
What proportion of the time is taken up from the computing the leverage scores and doing the sketching versus doing the integration afterwards?
Yeah, so the integration steps is depends how many batches you have, right? When you have hundreds of batches, even though they are small, that still takes a long time. Then the leverage score calculations mainly depends on the input of your initial matrix. So it really depends how the structure of the data look like. For example, like the mouse embryo data, we don't do any batch correction. We just sketch and project them. Yeah. And yeah. And you can see this just our sketch integration based on the leverage score. It's a framework. We can analyze millions of cells. It's just the leverage score sample can get the representative cells and then maintain the gene covariance matrix of the original data. And right now it's scalable to analyze tens of million cells. Okay, the last example, I want to show the case that we can combine our bridge integration and our sketch integration together to analyze how to analyze the data. So first, we build a single-cell RNA-seq reference of the PBMC. We know, right, we have good PBMC, but there are also a lot of PBMC publicly available. Then we just try to integrate all of them here. So we have a 3 million PBMC single-cell RNA-seq reference. And there's a UK consortium called Combat. They also have those different CyTOF data across around 5 million CyTOF data. Then we can use a site-seq, a PBMC site-seq in the middle as a bridge, and to project this CyTOF data onto this RNA space. And right, so that's the result. But to validate, right, if we map them properly, then we just do a heat map to check the marker genes of those cell types. Here we do the pseudo-bulk heat map. That means each column is just we aggregate the individuals and from their cell types. So that means each column here is not just one cell, but it's just one individual from the CyTOF data. In the CyTOF data, they measure both the surface protein and the intracellular protein. We only use the surface protein combined with a site-seq to do this bridge integration. Then here's general the marker genes, but here I want to highlight some hard annotated populations. For example, like this Treg, it's clearly show this transcription factor for T3 based on the integration only using the surface marker. And also in the mate cells, we see clearly they are the granzyme B deleted, and which is also reported before. The most interesting is those IOC that we annotate them independent from the original CyTOF data. And here we can see the correct immunophenotype of those IOC, right? The CD161 positive, CD25 positive, and CD56 negative. So that to prove that, okay, this is the correct annotation that we have. Right, so you can see in our Seurat 5 variants, we have all those examples like the cross-modality bridge integration and sketch clustering and the sketch integration. And beyond that, we also work on some new infrastructure because Seurat 5, not just those methods, we also have a new different object structures. Here, mainly, we just introduce some new concept called layers, and that will be more easy for users to do this scalable integration. And also, Seurat 5 is compatible with BP cells. That is on this computing infrastructure developed from Greenleaf's lab, and it's called BP cells. It can do a lot of those computing on disk. It can queue the operation there. That means instead of we store all the data in the sparse matrix, we can also store the matrix in the BP cells. That will make the Seurat object much, much smaller because all the things on the disk. Yeah. And the sketch integration, everything there is compiled with the BP cell matrix there. Then just in the end, so just thank Rahul for the guidance and make this project possible. And thank you for all our lab members, their endless help and support, and our Seurat team, right, to maintain the package and to make that available. And our Seurat 5 is very close to prime now. But right now you can still install Seurat 5 in our GitHub pages. Yeah, thank you.
So yeah, we have time for a couple of questions now, and then we'll break, then convene for a kind of informal discussion.
Hi, thank you for the talk. I had a question regarding the integration part. So when I think about multiome data, I kind of think about how like attack and RNA are different. So intuitively, like given two cells with similar attack profiles, they can have kind of different RNA expression profiles because of maybe transcriptional regulation and stuff. And I believe Xingyi gave an example where like if you just cluster based on attack and you just cluster based on RNA, you kind of get different clusters, suggesting that the nearest neighbor graphs on just attack versus just RNA are quite different. So my question is kind of, is that a problem when you're computing your like bridge cell representation?
Yes, thank you.
And how do you know when you've come up with a good enough one to kind of overcome that?
Yes, thank you. Right, like the question. Right, so that depends how do we process our bridge data, the multiome data. Right, if we only generate the cell graph using the RNA-seq in the multiome data, yes, we will lose all the attack-seq signal, those unique signals. But like I mentioned, the thing is we get the graph from the multimodal data is that we use our weighted nearest neighbor graph. That is one way, we try to capture the signal from both modalities. That means if you look at the cluster in those WN graph there, the cluster is not just based on single modalities, it's based on two modalities. Because we get those modality weights, we say, okay, for each individual cell, which modality have more information? If they have more, we're going to use that modality to generate those clusters. Then those things can maintain and guarantee that we are not going to lose those information for the tag specific or the RNA specific.
Thanks for the great talk. Just have a quick question about the sketching method that you mentioned. The example that you use is a sort of mixed cell type. Would that be a problem if I use a more like heterogeneous cell type, like if I just like use cell cultural samples where I only have T cells, for example?
So you mean just across different T cells, or you mean T cell with the PBMC, or?
Just T cell by itself. Like if I just take T cell, do cell culturing, and then do sequencing, so I only have T cell by itself. So I won't have sort of different cell types in a mixture. So would that be an issue if I do like using Seurat 5?
Yeah, I think it depends. For example, right, if you do a large-scale T cell perturbation experiment, right, there are a lot of in vitro T cells. But the thing is, right, the perturbation, they also provide the signal. So, right, in theory, you can also do the sketching, integration stuff, or on the perturb-seq, because when the perturbation have strong signal, you have the big weight in the gene covariance matrix of that data, you can also capture that. So that is not a big problem. It's mainly about those cells if they have the unique variance, unique signal in this data set.
Would it be accurate to say that in the case where you have a lot of cells but no unique variation, then you can take any?
Yes, then you can uniformly sample them.